Well, in spite of the traffic issues, our next speaker has made it, and I'd like to welcome now Mene Pangalosh, who is the Executive Vice President at AstraZeneca and based in Cambridge. And so I'm sure you'll give a nice welcome to Mene.
Thank you. Thank you very much, and apologies for being late. I thought Cambridge traffic was bad. London traffic was equally bad. So anyway, it's a pleasure to be here, and what I hope I can do is give you a flavour of not just the power of genomics, you heard it from the previous speaker, but actually show you in applied terms what it means in terms of potential treatment care and how it transforms the care of a number of different diseases. What I'll do is just give you a little bit of an introduction, first of all, into some of the approach that we take within AstraZeneca, but I think this is true across our industry, to give you sort of a grounding in terms of some of the challenges that we have faced, then give you some case studies that highlight examples of using personalised healthcare and genomics in oncology, and then try and show you how we're starting to broaden that out beyond oncology to impact a variety of disease areas. Let's start first of all with why this is important. So the good news is, as an industry, we're getting better. This graph here shows you the number of FDA approvals over the past couple of decades. And you can see that 2014, 2015, if you focus on the sort of dark blue bars, we've had record numbers of approvals, over 40 FDA approvals. And what's nice is that the percentages in terms of the number of drugs that are submitted to the FDA late stage development for filing have gone up in terms of that approval rate. So we're up in the sort of 90% range. When a drug is filed now, past 2 years, the success rate in actually getting that drug approved has increased. Now that's actually quite a dramatic change. One of the reasons why we're seeing, I think, an increase in our success rates is because we're understanding the disease that we work on better, the quality of the files is better, and we're using technologies like genomics and biomarkers, patient stratification, to create better files and a better definition of the patient population that's most likely to respond to that particular therapy. But we should put this into the context of our total success rates. When you look at the success rates from first in man to filing, we still fail 90% of the time. So just to put that into context for you, so every time a drug goes into man, you go through various phases of development, you get to the end, you succeed 1 out of 10. That's when you're doing it well. The industry average is actually around a 4% success rate. So we still have to do much, much better in terms of drug development. Now when I joined AstraZeneca, we did a piece of work to look at all of the data that we generated from 2005 to 2010. And the one underlying message that came from this piece of work to understand how we made decisions, why we made decisions, what the outcome of those decisions was in terms of the success or failure of a project, was that you have to focus on quality, not quantity, not the number of projects that you have, but how well you understand that program, that patient area, that disease area, that pathway that you're excited about, the molecule that you have. And you can't industrialize science or R&D. You have to do this project by project and be very thoughtful about each and every project that you run. Now as a consequence of that, we built a framework. This was published in Nature Reviews Drug Discovery about a year or so ago. I'm not going to go through all of this data, but the 5R framework is quite simple, quite intuitive, and all of our scientists now use it to think about when they start a program, what are the key measures or key attributes of a program that are important in determining actually its future success. I'm going to point to 2 of these and talk about them a little bit more during the course of the next 15 minutes or so. The first one is right target. Is really understanding the pathophysiology of the disease that you're trying to treat, the driver of that disease, and then the way that you're trying to modulate that pathway, and constantly challenging your scientific hypothesis, so you're not constantly trying to prove yourself to be right, but you're trying to work out whether your hypothesis is right or wrong. And you have to do that all the way through research and then all the way through development as well. And it's going to be a circle that continues to loop back from the clinic to the research lab, from the research lab to the clinic, and it's a cyclical thing, but you have to have that rigor, that curiosity, that truth-seeking behavior. The second piece that's incredibly important that I'll talk about is this R called right patient. And that's defining the patient population that is most likely to respond to your particular agent. Because if your drug doesn't work in that population, it's not going to work in a more heterogeneous, large population. So doing those experiments early, defining what the most responsive patient population is going to be, and having a strategy for getting to that population is very, very important. I hope I'll show you with some examples of the difference that makes. So as a consequence of that, we've actually embedded personalized healthcare, patient stratification, right at the beginning of when we start a project. So when a team comes forward with an idea, and I want to treat this particular target in cancer, or this particular target in asthma, or in diabetes, one of the things that teams have to think about is how are you going to try and stratify your diabetes population to give you the most responsive patient set for that particular mechanism. And as a consequence of that, when you look at our pipeline, and every green dot is a program that's got a personalized healthcare strategy, you can see that over 80% of our pipeline now, even early on in development, has got a strategy for trying to define what that population is. Okay, so let me give you some examples. That's kind of a high level what we're trying to do, and you heard from the previous speaker the power of genomics. Let me talk to you a little bit about two particular types of cancer. The first one is a drug called osimertinib, which used to be called AZD9291, for non-small cell lung cancer. Now, the history of lung cancer is actually quite remarkable, and it shows again what the power of genomics and the advent of next generation sequencing has done in terms of our ability to delineate drivers of disease. So pre-1980s, cancers would have been diagnosed by a pathologist. You would have had adenocarcinoma or squamous, large cell. You would have been diagnosed by pathology only. The first genetic driver of non-small cell lung cancer, KRAS, was identified in the mid-1980s. That was the first oncogenic driver. The second driver was identified in the mid-2000s. EGFR receptor, that's the epidermal growth factor receptor, a receptor that's constitutively activated in the cells, and it causes them to proliferate and cause the cancers to grow in your body. But look at what's happened 2012 and beyond. You can see all of the different drivers of non-small cell lung cancer that have been defined, and actually this is very out of date. There's at least another 10 genes or so that have been identified. And each one of those segments is a segment that you can go after with a very specific mechanism. And so this is the power of personalized healthcare, because now you can sort of define a patient population that is much more likely to respond to a particular targeted therapy using genetics. So we've talked about the EGFR receptor. There was a couple of drugs that were developed to target, actually several drugs that have been developed to target this EGFR activated mutation. One of them is a drug called gefitinib, and this is a study that it was actually done by AstraZeneca. The graph on the left-hand side shows you the effects of gefitinib in a non-selected patient population. So if you compare the orange curve, which is traditional chemotherapy, to gefitinib, the targeted agent, you can see that in the progression-free survival, as you go down the line, basically your tumor is progressing. You can see there's very little separation between those curves on the left-hand side. If you look at the curve on the right-hand side and you focus on the two purple lines, the solid line and the dotted line. The solid line is the EGFR activated population, the population of patients that has an EGFR mutation. The dotted line is a population that doesn't have an EGFR activating mutation. And you can now see how the curves separate in terms of their response to the drug and their progression-free survival. This is how you want to be doing your studies, because in this population now, we see a very dramatic impact on patient well-being and response to the drug, because we've got a mechanism that's targeting a specific driver of disease. But cancers are very smart, and patients develop resistance. We're not curing the patient here, even if they've got an EGFR activated mutation. We're seeing the disease is still progressing, it's just progressing more slowly. So one of the things that we've been very keen to do is to understand those resistance pathways, again, using genomics and our understanding of right target, the disease, and then develop new agents to start to address that resistance, to see if we can make lung cancer more of a chronic illness rather than a terminal illness. So, the EGFR activating mutation, this is actually the catalytic binding site of the EGFR receptor. This is where drugs like gefitinib bind, and actually you can see gefitinib binding into this catalytic site. And notice the small residue there on the top right-hand side, which is called T790. That's a threonine amino acid, number 790. And what happens in around 70% of patients that develop resistance to drugs like gefitinib is a single amino acid change. And you can just see that change. If I click backwards and forwards a little bit, you can see that change happening in the catalytic site of the receptor. And that single amino acid change from a threonine to methionine means that gefitinib is now kicked out of the active site of the enzyme, no longer able to block the activity of the receptor. The cancer develops resistance and continues to grow. So by understanding the biology behind this resistance, our chemistry teams can now develop a drug that actually binds into this new activating mutation and blocks the original EGFR mutation, but also now this new T790M mutation. So really deep understanding of the science and how to go after that particular resistance mechanism with a new molecule. And of course, when this drug went into the clinic, just over 3 years ago, the very first patient that was dosed responded. And this is the waterfall plot of all the patients that we've tested in the early stages of development. Just orientate on the graph. Horizontal line, time 0 is sort of at the starting point of treatment. If you fall below the line, your tumor is shrinking. If you're above the line, your tumor is growing. And the great thing about data like this is you don't need a statistician to tell you whether your drug works or not. Every dose in pretty much every patient that had a T790M activating mutation worked in this patient population. So this program was able to move super fast through development, through phase 1/2, and ultimately to late stage, and actually was approved in less than 4 years, less than 3.5 years actually, from the first time in man to launch, approval by the FDA, which is the power of genomics, the power of defining the right patient population and understanding the mechanism of the disease that you're trying to treat. Now what's also exciting here, and we heard it talked about later, is we're also the first company to launch actually a circulating tumor DNA diagnostic with our partners. And why is that important? It's not just important for early detection. It's actually important because many lung cancer patients can't have a biopsy. Their tumor is either inaccessible or for whatever reason the patients can't undergo the complexities of the biopsy to access tissue from the lung. So when you have a circulating tumor DNA diagnostic, it means that you can test a patient with a blood sample and then you can detect that T790M mutation or the original EGFR activating mutation, and they can get access to the right drug without having to have a biopsy. So it opens up the opportunity. It's obviously a lot cheaper than having a biopsy as well. So that's an example in lung cancer. The other important thing, and I talked about right target, is that we didn't stop there. As I said to you previously, when we're in research, when we're in development, we have to continue to try and understand the disease. 9291 doesn't cure non-small cell lung cancer. It prolongs the benefit for patients. People will develop resistance to 9291 as well. And so one of the things that our scientists were doing by taking samples from the patients that were in the clinical trials is starting to understand what are the resistance mechanisms to osimertinib. And we found 3 key drivers of resistance to osimertinib 9291. The first one was the amplification of a gene called c-Met, another known oncogenic driver present in a number of different tumor types. And the good news was that we had a c-Met inhibitor also in early development, so we're able to combine now 9291 with our c-Met inhibitor 6094. The diagram on the picture on the right shows you a lady that had an EGFR activating mutation, had become insensitive to EGFR inhibitors, and then we added our c-Met inhibitor on top of her therapy, and her tumor started to respond and to disappear. This is a woman in her early 30s, had a very poor prognosis, 3 to 6 months to live, now is on an EGFR inhibitor and a MET inhibitor, and her tumours have melted away and she's doing well. You can see her lymph nodes there, how they've responded to the two therapies combined. The second mechanism identified was MEK activation, another pathway that's very well understood to be important in cell proliferation. Good news again was that we had a MEK inhibitor in clinical development actually for KRAS non-small cell lung cancer. And so again, we've combined those two therapies, and this is actually some data from some preclinical experiments where we showed the impact of combining 9291 or osimertinib with our MEK inhibitor. So bottom left panel, you can see pre-treated animals. You can see the orange arrow pointing to the tumor. If you move along to the osimertinib treated arm, you can see that after 8 weeks of treatment with osimertinib, the tumor starts to shrink in this animal. But you can see that after 12 weeks, it's starting to grow back. Now, when you add the MEK inhibitor on top of that, you can start to see the tumor again regress and respond to the combination treatment. So we now have another study ongoing in the clinic where we pull out the patients that are developing resistance through activation of their MEK pathway and treat them in combination with our MEK inhibitor. Finally, we identified a new mutation that we published in Nature Medicine at the end of last year. And that's another mutation, the catalytic site of the enzyme, which is a cysteine residue, about 0.7 amino acid residue 797, converting to a serine residue. And so our chemists now are looking at the changes in the catalytic site and trying to develop a molecule that addresses that particular resistance mechanism as well. And we heard the previous speaker talk about gene editing and the power of CRISPR. And this is something that actually is really transforming how we do research in the labs, because we were able to create, using gene editing, the tools to enable our scientists to move on that new activating mutation at a pace that we could never have done previously. So in a matter of a couple of months, we were able to make these cell lines that contained not just the EGFR activating mutations, not just the T790M mutation, but also the C79S mutation. So a cell line that had basically all three mutations in a tumor cell line. You can see it at the bottom hand, like all edited in one go, using this fantastic technology. It enables you to basically manipulate whichever gene you want, however you want, in a very, very rapid and efficient manner. And that now gives us a screening tool that we can use to develop the next generation of molecule that hopefully will again confer benefit to patients that have that particular C79S mutation. So that gives you a case study in lung cancer. Let me speak for a moment about DNA damage response, because it's another really good example of how genomics, personalized healthcare, is transforming patient care in a variety of different tumor types. So, as you know, tumors divide rapidly, and as a consequence of dividing rapidly, they also have a lot of errors and mutations introduced into their genetic coding. And as a consequence of that, they become quite dependent on certain DNA repair pathways. And so the best known example that you will probably know is the BRCA genes. If you have a BRCA1 or a BRCA2 mutation, if you have a breast cancer sufferer or an ovarian cancer sufferer, those particular tumors are very dependent on single-strand DNA repair mechanisms, and in particular, an enzyme called PARP. So if you take a BRCA1, BRCA2 positive patient, and you treat them with a PARP inhibitor, the hypothesis would be that they would be more sensitive to that particular therapy, and you'd specifically kill their tumor cells and not their healthy tissue. And then there are other DNA damage repair pathways like ATM, ATR, target different tumor types and different potential patient populations. So if I just focus initially on PARP, this is the impact of a BRCA mutation on a PARP inhibitor. This is actually in a selected ovarian cancer population. Dark blue line is ovarian cancer patients that are BRCA positive. Light blue line is ovarian cancer patients that are BRCA negative. And again, you can see the separation in progression-free survival. Median changes of 4 months, from 4 months in the placebo-treated arms to 11.2 months in the treated arms. So a very, very dramatic change in response rate. Sorry, this is the 2 lines of placebo versus olaparib, not in a BRCA-selected population. So this is a drug, again, that's having a very dramatic effect in BRCA positive patients, and it highlights the power of personalised healthcare. When you go into an unselected population, it's less effective. Now, what's really exciting, though, is how you combine this with other agents. So I showed you the number of different enzymes and pathways that are important in DNA damage response. There's another pathway or an enzyme called WEE1, which blocks the activation or the phosphorylation of 2 checkpoints, CDK1 and CDK2. And by blocking these 2 checkpoints, it drives cells, it pushes cells through mitosis. It actually forces the cells to divide. And as a consequence of dividing too fast, not having time to correct their genome, it actually causes genomic instability and mitotic catastrophe and apoptosis. So in this particular study, this is actually looking at ovarian cancer patients that were resistant to chemotherapy, to platinum therapy. Their prognosis is incredibly poor. So this is a platinum-resistant ovarian cancer patient population. Their normal response rate to chemotherapy would be about 10%. Their duration of therapy, in terms of how long they can stay on therapy before their tumor would progress, is normally around 3 to 6 months. And you can see the benefits here of a WEE1 inhibitor. So this is in that particular population, we see around a 40% response rate compared to a 10% response rate with chemotherapy. And importantly, the level of response that we see, the depth of the response, we see some women responding with 90%, 100% of their tumors shrinking. So almost to the level where they can't be detected in the scanner. And the other really exciting thing is the duration of response. We have women that have been on drug now for over 2 years in a population that would normally respond for 3 to 6 months. So really exciting data, and this program should be moving into late-stage development later on this year. But even more exciting was the data that we generated when we combined a PARP inhibitor with a WEE1 inhibitor. And this is in a preclinical model. This is data we generated last year. You can see in the blue line, olaparib, our PARP inhibitor. You can see in the green line and the orange line, our WEE1 inhibitor, both given as monotherapy in this animal model. We stopped dosing the drugs at day 28. When we combine, in the red line, if you look at the red line, we combine olaparib with our WEE1 inhibitor, and we stop dosing the drug at day 28, the tumors don't come back. So you can see that tumors growing, if they're growing, then basically the line's going up the graph. You can see the red line basically starts to grow, shrinks, but doesn't come back after you stop dosing. So we basically completely eliminated tumor growth in this particular animal model. I'm not proposing we're curing the cancer here, but if we can see this type of synergy in the clinic, combination of olaparib with WEE1, we should see a very dramatic benefit in a number of different tumor types that are sensitive to DNA damage response. So very, very exciting data. Now, finally, just to continue the last bit on oncology, we've talked about the power of genomics, and it is incredibly important and really transforming how we can define the genetics drivers of disease for any particular patient. One of the things that we're seeing more and more of is these things that are called basket trials. And they're incredibly important, incredibly powerful, because if you think about that lung cancer picture I showed you and how many different genetic drivers of lung cancer there are now, each one of those segments is actually quite small. 1% of the lung cancer population, 0.5%, 5%. So if you're doing a clinical trial and you're trying to screen for patients with a very rare mutation, you have to screen 100 patients to find one that might be sensitive to your study. So what we've done, working with groups like Cancer Research UK and actually other companies as well, is we've actually put all of our molecules into a basket so that when a patient comes in, is sequenced and is identified to have a particular driver of disease, the physician has access not just to one molecule, but 20 molecules. So they can actually pick and choose as they define the patient, the driver for that particular patient, and it makes the trial much more effective for signal searching and actually for accessing patients more quickly. It's better for the patient, it's better for the physician, and it's actually better for us because it speeds our ability to find signals in the right patient population. So these basket trials are happening more and more and more, and I think are really having an impact. And the only reason they're happening is because next generation sequencing is that much easier and that much faster and that much cheaper now that it's actually feasible. Okay, so I've talked about personalized healthcare, patient stratification, genomics in cancer. What about beyond cancer? What are we doing in other therapy areas? Well, the truth is that, you know, oncologists in the cancer field, I think, has actually led the way in terms of how we've done this. But I think other fields, other areas are trying to catch up and trying to do their particular piece to define their patient segments in a much better way. So I'll give you a couple of examples. First one is Alzheimer's. I'm a neuroscientist by training. I've worked on Alzheimer's disease for many, many years. I actually ran some of the first immunotherapy programs for the treatment of Alzheimer's disease that unfortunately failed. One of the reasons why they failed is because about 40% of the patients that we recruited into our trials didn't actually have Alzheimer's disease. That's kind of a problem when you're trying to develop an Alzheimer's drug. So we've become, again, much more careful at selecting Alzheimer's patients through a number of different ways. We measure now proteins in the cerebrospinal fluid of the patients to make sure that they have the protein biomarkers that are in keeping with the diagnosis of Alzheimer's disease. We use PET imaging to look for markers such as beta amyloid, as you can see in that image there, that differentiate an Alzheimer's patient from a healthy patient or a patient that has some other form of dementia. We use genetics, ApoE genotype, family history, as well as monitoring cognitive decline over time. So the criteria now for getting into an Alzheimer's trial much more rigorous, and I think the trials that are enrolling right now are much more likely to succeed because they have a much better selected patient population. They're also, as a consequence, going into an earlier patient population, which makes it hopefully more likely to see a benefit. In asthma, we've seen some of the first biologics approved in a subset of asthmatic patients that have highest eosinophil counts in their blood. This is data from one of our studies targeting an IL-5 receptor. This is an anti-IL-5 receptor monoclonal antibody. We're looking at exacerbation rates, and along the x-axis you can see increasing blood eosinophil counts. And you can see that as you have more eosinophils in your blood, so you respond better to this particular therapy, and GSK has generated very similar data with mepolizumab, which is their monoclonal antibody to IL-5. So as a consequence of this, the stratification strategy for this particular mechanism is eosinophil counts. So will there be a cutoff above which you'll be able to take this medicine and you're more likely to respond to this medicine, making more effective, making the benefit risk better, and ultimately making more attractive for payers, physicians, and patients. In gout, we don't often hear people talking about gout, a high unmet medical need. There haven't been any new therapies for gout for some time. We just had one approved at the end of last year. But there's no really good point of care diagnostic to determine people's uric acid levels. So we've been working with a partner, Nova Biomedica, to develop a measure of uric acid in the blood that's easy, can be done by a primary care physician to determine not just the level of uric acid, but also the response to the medicine to make sure we get the dose right. And then finally, just to talk about next generation sequencing at all, and obviously the previous speaker covered this in a lot more elegantly than I will, but with the advent and the lowering of the costs of sequencing, we're seeing more and more population studies being done by Genomics England. We have studies actually running all over the world. The challenge actually for all of this isn't going to be generating the genetic data. It's going to be integrating and interpreting the data. It's going to be thinking about the questions that you have to ask and then pulling out the needles from the haystack. We're going to be generating so much. If you saw the capacity, 1.5 million genomes per year is the global capacity that we have around the world. That's petabytes and petabytes of data. It's so much data you can't even imagine, actually. And the biggest challenge is going to be having the bioinformaticians, the statisticians, the scientists that can really interrogate this and make sense of it all. And actually in the UK, we don't have enough of those people. So we're also going to have to do some training to get people to really be able to handle, manage, interrogate and make sense of all of this really important data that we're generating, because it's going to be very, very powerful potentially and very, very important and can transform healthcare. Find rare variants, find rare polymorphisms that will enable us to create better tailored medicines for particular patient populations. Okay, with that, I hope I've given you a taste of what we're doing, the future, the power of genomics and personalized healthcare, and the impact that it can have on defining the right drug for the right patient and ultimately giving the best benefit and the best outcome. Thank you very much.
Thank you, Mene. That was really inspirational and very exciting, and I think you'll all agree that the two presenters we've had this morning have just really reinforced the fact that this really is a genomic revolution. We've been through industrial revolutions, technology revolutions, but this is really quite a genomic revolution. So we have a couple of minutes for one question. If anyone has a question, could they put their hand up and we have a roaming mic? There's one here, yes, this gentleman.
Hi, great talk, thanks.
Thank you.
My name is Chris from Horizon Story. I just wondered, given the incredible efficacy that you're seeing with some of these combination therapies, I wondered how, or whether that's raising some questions about the ethics of giving placebos and how you or maybe the regulatory bodies are kind of dealing with that issue of moving quickly once you see such fantastic results.
Yes, so more often than not in cancer trials, you're not actually giving placebo, you're either doing single arms or you're doing standard of care versus the new therapy, or a combination of standard of care and the new therapy. So you're absolutely right. So again, depending on the disease area, depending on the efficacy of current therapies, you have to obviously take into account whether you do placebo-controlled trials or single-arm trials.
So let's give a very big thank you to Mene. Thank you very much indeed.